Good morning, friends. Today we are going to discuss second part of cell cycle and cell division of 11th standard. In this session, we will discuss about meiosis, meiosis 1, meiosis 2 and significance of meiosis. The term meiosis was introduced by J.B. Farme and Murray in the year 1905. Meiosis is confined to certain cells and that cells are called a germ cell. They are for producing gametes in sexually reproducing organisms. In higher plants, meiosis also forms asexual reproductive bodies called as spores. Many stages of meiosis are similar to mitosis. Meiosis is divided into meiosis 1 and meiosis 2, but only a single cycle of DNA replication takes place. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis includes two successive divisions without DNA replication between them. Synapsis, that is pairing of homologous chromosome, crossing over of segments between non-sister chromatids of synapsed chromosomes and formation of chiasmata, segregation of homologous chromosome and separation of sister chromatids. At the end of meiosis, 4 haploid cells are formed. In the given figure, you can observe the different stages of meiosis, that is meiosis 1 and meiosis 2. Meiosis 1 is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1, whereas meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. All these stages we are going to discuss here. First, it is about prophase 1 of meiosis 1. The prophase 1 of meiosis 1 is more elaborate and lasts longer than the prophase of mitosis. It takes more than 90% of the time required for completion of meiosis. Unlike prophase of mitosis, in prophase 1 of meiosis 1, DNA is exchanged between homologous chromosomes and there is formation of new combination of genes due to crossing over. In prophase 1, the paired chromosomes are called bivalents or tetrads. Non-sister chromatids may cross over at points called chiasmata. Prophase 1 is again subdivided into 5 substages on the basis of chromosomal behavior. These 5 substages are leptotin or leptotina, zygotin or zygonyma, pachytin or pachynyma, diplotin or diplonyma and diakinesis. In the figure, it is of prophase 1. You may observe here 2 pair of homologous chromosomes. Now, we will discuss in detail about the different subphases of prophase 1. First, we will learn about leptotin. Leptotin is the Greek word which means thin threads. Here, chromosomes differentiate from chromatin fibers. They are very long and fine filament. Chromosomes begin to condense and the two sister chromatids are still so tightly bound, indistinguishable and are indistinct. Here you may observe the figure which show leptotin stage of prophase 1. Now it is about zygotin. Zygotin or zygonyma means paired thread. The homologous chromosomes come to lie side by side in pairs. This pairing of homologous chromosomes is called synapsis. Pairing of homologous chromosomes is in such a way that the corresponding ends and all the corresponding genes lie exactly opposite to each other. Synapsis or pairing of homologous chromosomes is followed by formation of a complex structure called synaptonymal complex. A pair of homologous chromosomes lying together is called as bivalent or a tetrad. In the given figure, you may observe the zygote stage of prophase 1. Here you can observe the pairing of homologous chromosomes with synaptonymal complex in between them. Next is about packeting. Packeting means thick thread. The synapsed chromosomes continue to become short and thick. The chromatids of each synapsed chromosome slightly separate and become visible. The two visible chromatids of a chromosome are called dyad and four homologous chromatids are called tetrad. The two chromatids of the same chromosomes are called sister chromatids and those of two homologous chromosomes are called non-sister chromatids. Packeting is characterized by appearance of recombination nodules. There is exchange of genetic material between this site where recombination nodules have formed. This recombination nodule is also called as chiasmata. Thus, in packeting, crossing over takes place between non-sister chromatids of homologous chromosome. In crossing over, enzyme recombinase is involved. After crossing over, there is recombination of genetic material at the end of packeting. So, here there is mutual exchange of the corresponding segments of non-sister chromatids of homologous chromosome. It takes place by breakage and reunion of chromatid segments. Here in this figure, you may observe packeting stage of prophase 1. Diplotin. Now it is about diplotin. Diplotin or diplonema means two thread. In this phase, the homologous chromosome starts separating. Synaptonymal complex degrades and the chromosome uncoil a bit. But the homologous chromosomes remain tightly bound at X-shaped structure called chiasmata. This is the identifying features to locate diplotin stage. It means that separation of homologous chromosome does not take place at the point of chiasmata. Indication of chiasmata shows that crossing over has taken place during packeting. In oocytes of some vertebrates, diplotin can last for months or years. In the given figure, you may observe diplotin as well as X-shaped structure that is called as chiasmata. Now it is about the final stage of meiosis 1 of prophase 1, that is diakinesis. The chromosome condense and chiasmata disappear, called as terminalization of chiasmata. There is appearance of spindle fibers and by the end of diakinesis, nucleoli disappear, nuclear membrane breaks down and it also disappears. Here it is the figure which shows about the diakinesis. In this figure, you may compare all the stages of prophase 1 of meiosis 1, that is leptotin, zygotin, packetin, diplotin and diakinesis. Now we will discuss about metaphase 1 of meiosis 1. Here the bivalent chromosomes become arranged in the plane of the equator, forming equatorial plate. The centromere of each chromosome is directed towards the opposite poles and the arms of the chromosomes face the equatorial plate. Meanwhile, the microtubule from the opposite poles of the spindle get attached to the pair of homologous chromosome. The figure shows about the metaphase 1 of meiosis 1. You can see here metaphase plate on which the chromosomes are arranged, and also you can observe the microtubules attached to the kinetochore or centromere. Now it is about anaphase 1 of meiosis 1. Here the homologous chromosome of each pair rather than the chromatids of a chromosome are separated. Thus each pole receive half the number of chromosomes or haploid set of chromosomes. Why? Because the chromosomes itself move to the opposite poles. So reduction in the number of chromosomes takes place. Here in the given figure, you may observe about the separation of homologous chromosomes, but sister chromatids remain attached. Now we will discuss about telophase 1. In telophase 1, the chromosomes arrived at the poles. Two daughter cells now has half the number of chromosomes. Spindle fibers now disappear. The chromosomes uncoil back into chromatin. Further, cytokinesis takes place to form two daughter cells. Now the cells may enter a period of rest known as interkinesis or interphase 2. There is reappearance of nuclear membrane and nucleolus. Here you can observe telophase 1 and cytokinesis 1. You may see here that chromosomes reach to opposite pole and cytokinesis begins, and the two new cells are produced, each containing half chromosome in each cell. Now let us discuss about meiosis 2. Meiosis 2 is similar to mitosis. At the end of meiosis 2, there is production of 4 haploid cells. It consists of 4 main steps, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. We are going to discuss here one by one. First we will discuss about prophase 2. Prophase 2 is very short. There is disappearance of nucleoli and the nuclear membrane. The chromosomes, each comprising 2 chromatids, become visible in the nucleus. There is shortening and thickening of the chromatids. Centrioles moves towards the polar regions and arrange the spindle fibers. Here you can see the prophase 2 of both the daughter cells. Now it is about metaphase 2. Here the centromeres contain 2 kinetochores that attach to the spindle fibers. There is alignment of chromosomes at the equator. The microtubules from opposite poles of the spindle get attached to the kinetochores of the sister chromatids. In this figure, you may observe the metaphase plate in both the cells, how the chromosomes are arranged. Now let us discuss what is happening in anaphase 2. In anaphase 2, centromeres get cleaved and spindle fibers attached to kinetochores to pull the sister chromatids apart. Now the sister chromatids are called sister chromosomes. You can observe here how separation of sister chromatids takes place in anaphase 2. Now it is about telophase 2. Here uncoiling and lengthening of chromosomes takes place. There is disappearance of a spindle and reappearance of nucleolus and nuclear membrane. Then cytokinesis takes place by cleavage or cell wall formation. So at the end of telophase 2, 4 daughter cells with haploid set of chromosomes are formed. Here you can observe telophase 2. It is about meiosis 2, where you can observe prophase 2, metaphase 2, anaphase 2 and telophase 2. Now, the last topic of today's session is about significance of meiosis. It is very important. It helps in maintenance of chromosome number. Meiosis produces gametes for sexual reproduction. Meiosis forms spores in plant for asexual reproduction. It helps in increasing genetic variability also. So, it leads to evolution and also it leads to mutation. So, this is all about today's session. Thank you. For any kind of doubts, please write your question in comment section.